Okay, let's unpack this. Imagine a system in your body that's been influencing your mood, pain, appetite, even how you react to stress for, well, millennia, really, long before we even knew it existed.
Exactly. We're talking about the deep biological reasons why cannabis has been used, medicinally and psychoactively for thousands of years.
But our actual scientific understanding of how it all works, that's remarkably recent. Today, we're diving deep into something truly foundational, the endocannabinoid system, or ECS.
And what's just, well, fascinating here is how this system made up of compounds our own bodies produce, specialized receptors they talk to, and the enzymes that build and break them down orchestrate such a huge range of vital actions,
It really is
from
vast.
stress regulation, pain perception, metabolism, inflammation. The ECS is like a master conductor. Our mission today is to give you, our listener, a really deep understanding of its intricate dance. We're aiming to get into the molecular mechanisms with the kind of precision elite professionals in this field would have.
Right. So we'll explore the players you might recognize, like THC and CBD from the cannabis plant, sure. But we'll also introduce you to their internal counterparts, the endocannabinoids, which our bodies make naturally.
And we'll uncover the truly remarkable processes for how our bodies create these crucial messengers, and then, just as importantly, how they precisely dismantle them.
Then we'll connect all that detail to the bigger picture. Why mastering the system is so critical for fields from, say, neuroscience all the way to advanced drug development. We think there'll be some real aha moments along the way.
So let's start right at the foundation. What is the endocannabinoid system? Think of it as maybe your body's most versatile internal regulator, sort of a highly responsive feedback loop working behind the scenes, constantly striving for balance, for harmony, across so many different functions.
That's a really good way to put it. At its core, the ECS is an endogenous signaling system. Endogenous meaning from within.
Yeah.
And when we break it down, there are really three main players, each one absolutely critical.
First, you've got the endogenous cannabinoids, often just called ECBs. These are unique lipid-based molecules. Fat-based molecules our own bodies produce naturally. They're specifically designed to bind to and activate cannabinoid receptors.
So our internal messengers?
Precisely. Second are the endocannabinoid receptors. These are specialized proteins, usually embedded in cell membranes all throughout the body. When ECBs or other cannabinoids like THC from cannabis bind to them, it's like a key turning in a lock. It triggers specific responses inside the cell, changing its activity.
Got it. Key and lock.
And third, and this is crucial, we have the enzymes. These are the molecular machines that control both the synthesis, the creation of these ECBs, exactly when and where they're needed, and also their rapid degradation, their breakdown.
Ah, the cleanup crew.
Exactly. It ensures the signal is precise, doesn't just linger forever. This dynamic, this on-demand control over making them and breaking them down, is what makes the ECS so incredibly responsive and adaptive. And what's truly remarkable is just how widespread it is. It's present across virtually every major organ system, playing a fundamental role in an incredibly diverse range of functions: stress, pain, metabolism, sleep, inflammation. The list goes on.
Okay, so if this system has its own internal messengers, let's meet the two main ones, the stars of the show, if you will. We're talking about anandamide, AEA, often called the bliss molecule, and 2-arachidonyl glycerol, or 2-AG.
That's right. These are the archetypal endocannabinoids, and they're fascinating signaling lipids derived from something called arachidonic acid, a fatty acid. Now, while they were first really studied heavily in the nervous system, it's vital for you to understand they're produced by cells throughout nearly every organ system in our bodies.
And I understand there's a bit of a challenge with how they move around the body. They're sort of oily.
Exactly. They're lipophilic fat-loving. Yeah. They don't mix well with water, which makes up most of our body fluids, like blood or the fluid surrounding brain cells. So getting around is tricky.
How do they manage it?
They cleverly associate with protein-binding partners. Essentially, they hitch a ride on molecules like fatty acid-binding proteins or albumin in the blood. These proteins act like tiny ferries, shuttling the ECBs through watery environments to where they need to go.
Okay, let's dive into anandamide, AEA, first.
The bliss molecule.
Yes, AEA, formerly N-arachidonyl-lethanolamine. It was the very first endocannabinoid discovered, and its name comes from the Sanskrit word ananda, meaning bliss or joy, which really hints at its impact on mood and well-being.
And structurally,
It's quite elegant, an arachidonic acid molecule linked, conjugated, to an ethanolamine group. Now, within the brain, AEA isn't stored up in little packages of vesicles like many classical neurotransmitters are. You know, those are released in big prepackaged bursts.
AEA is different. It's synthesized and mobilized on demand. That means it's precisely when and where it's needed, in a more continuous, though variable fashion. This on-demand nature is absolutely fundamental to its role as a rapid localized signal.
And how does it interact with the receptors?
Pharmacologically, AEA is what we call a high-affinity agonist at cannabinoid receptors. It binds very strongly, even at tiny, low nanomolar concentrations. However, and this is a critical distinction, it's a low-efficacy agonist.
Low efficacy.
I mean, it only partially activates the receptors. It never pushes them to their maximum possible response, so it's partial agonist. This subtlety is key to understanding its physiological role. Interestingly, at slightly higher concentrations, still potentially physiological in certain situations, AEA can also activate another type of receptor called TRPV1.
TRPV1, isn't that involved in pain and heat?
Precisely. The same ones activated by capsaicin in chili peppers. Now, when we measure AEA levels, the overall amount in brain tissue, the bulk level, is very low picomoles per gram. But, and this is a key insight for professionals, studies using microdialysis, which measures the levels in the fluid between cells where the signaling actually happens.
The active zone.
Exactly. Those studies show extracellular AEA in the low nanomolar range. This big difference between the total amount in the tissue and the active amount outside the cells really underscores its role as a rapidly made, locally acting messenger that doesn't just flood the whole system, it's very targeted.
And in the blood?
Similar story, low picomole per milliliter levels circulating. Though, honestly, the precise tissue source of that circulating AEA, whether it's coming from blood cells, the lining of blood vessels, liver, fat cells, that's still an active area of research. We don't fully know yet.
Okay, so AEA is the subtle, localized bliss molecule. How does its partner 2-arachidonyl glycerol, 2-AG, stack up?
All right, 2-AG.
Yeah.
The second ECB discovered. Structurally, it's also based on arachidonic acid, but this time it's conjugated to a glycerol group.
Oh.
But here's where things get really quantitatively different and quite intriguing.
How so?
Well, within the brain, there's strong evidence that 2-AG isn't just made on demand. It seems it can actually be stored, held in reserve, within specialized membrane structures called lipid rafts.
Yeah.
And when you compare their overall concentrations in brain tissue, 2-AG is present in the low nanomole per gram range. That's roughly 1,000 times higher than AEA's bulk concentration.
Wow, 1,000-fold.
Huge difference, right? Yet, similar to AEA, when we use microdialysis to measure the extracellular levels, the active signaling levels, 2-AG concentrations are only about 2 to 5 times higher than AEA's, still in that low nanomolar range.
So most of it isn't actually floating around signaling.
Exactly. This leads to this fascinating prediction, the 90% hypothesis, that about 90% of the 2-AG in the brain exists as this bulk membrane-associated pool. And this isn't just inert storage. This large pool of membrane-bound 2-AG is thought to function as a crucial reservoir for arachidonic acid itself.
The building block.
A building block, yes, which is a required substrate for forming neuroinflammatory prostaglandins, those signaling molecules involved in inflammation in the brain. This was a critical insight linking 2-AG directly to inflammatory responses.
And its action at the receptors. Different from AEA.
Very different in one key way. In contrast to AEA's partial agonism, 2-AG is a full agonist at cannabinoid receptors. It can elicit a maximal cellular response. It binds with nanomolar affinity, maybe slightly less potently than AEA, but it gives the full kick, so to speak. In the blood, circulating 2-AG levels are also low, similar to AEA, in the lower picomole per milliliter range. And again, its precise tissue source isn't fully nailed down.
Okay, so AEA and 2-AG are clearly the heavy hitters here. But is that the whole story, or is there maybe a wider family, a kind of endocannabinoidome of related molecules that also play a role?
That's a really sharp question.
Yeah.
And yes, while AEA and 2-AG are absolutely the most well-established, the most physiologically relevant ECBs we know of, the concept of a broader endocannabinome is definitely something researchers explore.
So what else might be in there?
Well, it suggests a wider family that could potentially include other related fatty acid acetylamides, various monoacyl glycerols, maybe even certain peptides. One molecule that's gotten a fair bit of attention is hemopressin. It's actually a fragment derived from hemoglobin's alpha chain.
From hemoglobin. Interesting.
Yeah, and it binds with high affinity to cannabinoid receptors. But unlike AEA and 2-AG, which activate the receptor, hemopressin seems to act as an inverse agonist.
Inverse agonist, meaning it turns the receptor off.
More than just blocking it, yes. It actively suppresses any baseline or constitutive activity the receptor might have, even without a signal. But, and this is really important, the actual physiological relevance of hemopressin in a living organism when and where it's made and released in a meaningful way, that's not yet well established.
So interesting, but maybe not a major player day to day.
That seemed to be the current consensus. Other molecules like virotamine and noliden ether have also been proposed as ECBs. But again, for these, it's just not clear if they're consistently made and released in vivo in a way that produces significant physiological effects. And that's precisely why, for really mastering the fundamentals of the ECS and its core functions, our deep dive will keep focusing primarily on AEA and 2AG. That's where the vast majority of our solid understanding of ECS physiology lies.
Okay, we've met these incredible internal messengers, but how does our body actually make them? It must be a pretty sophisticated process to get that on-demand timing right. Uncovering these pathways tells us exactly when and where the body decides to send these signals, doesn't it?
Absolutely. You can think of it like a very finely tuned molecular assembly line, constantly building these messengers exactly when and where they're needed for that rapid response. Let's start with anandamide, AEA, synthesis. Its creation seems to involve several, maybe somewhat redundant pathways, but they all seem to begin with a common precursor molecule called N-arachidinoyl phosphatidylethanolamin, or NARPE for short.
NARPE.
And NARPE itself is formed through a calcium-dependent enzymatic process. An enzyme called an N-acyltransferase basically takes arachidonic acid, often from a phospholipid in the cell membrane, and links it to ethanolamine, which comes from another phospholipid, phosphatidylethanolamin.
Okay, so NARPE is the starting point. Then what?
From NARPE, there seem to be three main routes to get to AEA. The first, and probably the best established pathway, involves an enzyme called NAPE-PLD. That stands for N-acylphosphatidylethanolamin specific phospholipase D. Quite a mouthful.
ANPEPLD. Got it.
NAPE-PLD directly snips NARPE, releasing AEA and another molecule called phosphatidic acid. And we know it's important because studies show that inhibiting NAPE-PLD can cut brain AEA levels by up to half.
Wow. Okay, so that's a major pathway. What are the others?
A second pathway involves NARPE being converted first to an intermediate called lysin-RBPE. This step can be done by a couple of enzymes, ABHD4 phospholipase A2. Then this lysin-RPE is further processed to something called GPAEA, which is finally converted to AEA by an enzyme called GDE1.
More steps in that one.
A bit more intricate, yes. And then there's the third pathway. Here, NARPE gets converted to a phospho-AEA by a phospholipase C-like enzyme. Then that phosphate group is snipped off by a tyrosine phosphatase called PTPN22, leaving you with active AEA. This particular pathway might be more important in certain immune cells, like macrophages, especially when they're dealing with inflammation. So different routes might be favored in different cell types or situations.
That's quite complex for AEA. What about 2AG? Is its synthesis pathway simpler?
For 2-arachidinoyl glycerol, 2AG synthesis, the process, while still involving multiple steps, really centers around one key type of enzyme, diacylglycerol lipase, or DAGL. There are two important forms. DAGLA, which is the main one doing the job in the central nervous system, and DAGLA, which handles 2AG production mostly in peripheral organs like the liver.
DAGL alpha and beta.
Right. And the typical pathway starts with the breakdown, the hydrolysis of a crucial membrane phospholipid called PIP2, that's SN2-arachidinoyl phosphatidylacetyl 4-vol-5-bisphosphate. This breakdown is done by an enzyme called phospholipase Go, PLC, and it generates diacylglycerol, or DAGA.
Okay, DAG.
And then DAG is converted to 2AG by that DAGL enzyme through hydrolysis. So it's still a precisely orchestrated assembly line, ensuring these messengers get built right when and where they're needed for those rapid local responses.
So we've got these messengers being built on demand. But if they're constantly being produced, how does the body stop the signal? How does it prevent overstimulation and make sure the message is brief and precise? There has to be a rapid clearing mechanism, right? A way to turn them off.
Precisely. Rapid degradation is absolutely vital. It's what ensures that on-demand nature of ECB signaling. It stops the receptors from being constantly bombarded and allows the system to reset and respond dynamically to new signals. Without efficient breakdown, the whole system would get overwhelmed.
Makes sense. So how is anandamide AEA cleared out?
For AEA degradation, the primary enzyme, the main player, is fatty acid amide hydrolase, or FAAH. FAAH is found pretty much everywhere throughout the body. It's a membrane protein usually associated with intracellular membranes specifically, the endoplasmic reticulum and mitochondria inside the cell.
And what does FAAH do?
Its job is to hydrolyze AEA, basically, chop it up, breaking it down into free arachidonic acid and ethanolamine. This effectively terminates AEA's signaling ability. Now, FAAH is considered constitutively active, meaning it's always working at some baseline level. But its expression and activity are also finely tuned, regulated by things like reproductive and metabolic hormones, and even through protein kinase A signaling pathways.
And where is it located within neurons?
Within neurons, FAAH tends to be found in the somatodendritic compartments, the main cell body and dendrites, where the neuron receives signals. How AEA gets from the membrane where it acts to the FAAH enzyme inside might involve those fatty acid binding proteins again, acting as little intracellular chaperones.
And how important is FAAH overall?
Hugely important. This is really significant for professionals. If you genetically delete FAAH in mice or block it with a drug, you get a large and sustained increase in AEA levels throughout the body. This proves it's the critical dominant enzyme controlling AEA levels.
And you mentioned something earlier about a human genetic variation related to FAAH.
Yes, this is fascinating for human pharmacology. There's a common single nucleotide polymorphism, a SNP, known as P129T in the human FAAH gene. People with this variant actually break down the FAAH protein faster.
So they have less FAAH enzyme around.
Exactly. Less FAAH enzyme means less AEA breakdown, which leads to naturally higher baseline levels of AEA signaling. This is a fantastic real-world example of how a small genetic difference can significantly impact an individual's endocannabinoid tone, and we'll definitely come back to what that means physiologically later.
Any other quirks with human FAH?
Well, humans actually have a second FAAH gene, FAAH2, which rodents don't have.
Yeah.
Its exact role isn't fully clear, but it hints at some human-specific regulation. Also, FAAH isn't totally specific to AEA. It can break down other related fatty acid amides and anisyltamines. So, inhibiting FAAH doesn't just raise AEA. It causes wider changes in lipid signaling. And finally, FAAH isn't the only way AEA is broken down. It can also be oxidized by the enzyme cyclooxygenase 2, COX2.
Inflammation enzyme.
That's the one. This reaction generates bioactive prostaglandin ethanolamides, adding yet another layer to AEA's metabolic fate.
Okay, so SAA is the main off switch for AEA. What about 2-AG? Does it have a similar dedicated enzyme?
Yes, it does. For 2-arachidonyl glycerol, 2-AG degradation, the primary enzyme is monoacylglycerol lipase, or MGL. And just like FAAH for AEA, if you genetically delete MGL or block it pharmacologically, you see a large sustained increase in 2-AG levels throughout the body. Unequivocally, MGL is the key player in controlling 2-AG concentrations.
And what does MGL break 2-AG down into?
MGL hydrolysis breaks 2-AG down into free arachidonic acid and glycerol. And this connects back to something crucial we discussed earlier. The precise control of both synthesis and degradation is what allows the ECS to function as this real-time dynamic signaling system. It responds rapidly and transiently rather than just flooding everything constantly.
Right. And you mentioned MGL's connection to inflammation.
Yes, this is really significant, especially in the brain. Remember, about 90% of brain 2-AG is thought to be in those membrane reservoirs holding arachidonic acid. In response to inflammation, AGL activity ramps up. It starts rapidly breaking down this membrane-associated 2-AG.
Releasing the arachidonic acid.
Yes, exactly. Liberating arachidonic acid, which is then quickly oxidized by COX2 to generate those neuroinflammatory prostaglandins. And research specifically pinpointed MGL located in astrocytes, the brain's support cells, not the neurons themselves, as mediating this generation of inflammatory molecules from 2-AG breakdown, a critical link in neuroinflammation.
Where is MGL found location-wise?
MGL is widely expressed, both in the nervous system and peripheral organs.
Yeah.
But interestingly, unlike FAAH, which is often somatodendritic, MGL in neurons is primarily localized to presynaptic compartments, the axon terminals.
Ah, so it cleans up 2-AG after it's done its job at the synapse.
That's the prevailing hypothesis, yes. It suggests MGL clears the signal precisely from the synapse, allowing the presynaptic neuron to reset. While some MGL is found floating in the cell cytosol, its association with membranes likely helps it efficiently hydrolyze 2-AG right where it's needed. And similar to FAAH, MGL isn't totally specific. It also hydrolyzes other monoacylglycerols. So inhibiting MGL causes widespread elevations in various lipid signaling species, not just 2-AG, which is important to remember when considering MGL inhibitors as drugs.
Is MGL the only enzyme breaking down 2-AG?
It's the dominant one by far. But other hydrolases like ABHD6 and ABHD12 can also contribute. ABHD6 is found in the somatodendritic compartment, suggesting it might regulate 2-AG before release, a different control point than MAG. ABHD12 seems to handle maybe around 9% of 2-AG breakdown in the brain and is particularly abundant in microglia, the brain's immune cells, hinting at a role there. Inhibiting these can affect 2-AG signaling, but it's generally much less impactful than blocking MAGL. And finally, like AEA, 2-AG can also be oxygenated by QX2, forming prostaglandin glycerols, but their physiological role is still being figured out.
Okay, so we've got these fascinating messengers, AEA and 2-AG, being precisely made and then rapidly broken down. But how do they actually exert their effects? This brings us squarely to the receptors, the targets where these molecules bind and initiate a response. Now, historically, figuring out how THC, the main psychoactive component in cannabis, actually worked was a real puzzle, wasn't it? Partly because it's so fat-loving.
That's absolutely right. Its lipophilic nature made it incredibly difficult for scientists for many years to pin down a specific receptor. It just seemed to dissolve into cell membranes. But then came the groundbreaking work from Alan Howlett and her colleagues back in 1990. They provided the first really convincing evidence that THC's actions were mediated by a specific type of receptor called a G-protein-coupled receptor, or GPCR.
How do they show that?
Through elegant experiments, they demonstrated that THC and related synthetic cannabinoids inhibited an enzyme called adenyl cyclase, and importantly, this inhibition happened via a pertussis toxin-sensitive GPCR. That sensitivity to pertussis toxin was a key signature of a specific family of GPCRs. This is a monumental
A
discovery.
real breakthrough.
Absolutely. And then using techniques like autoradiographic mapping, which essentially lets you see where receptors are by labeling them, researchers confirmed that this newly found cannabinoid receptor was present in high levels across many brain regions in a pattern that perfectly matched the known psychotropic effects of THC. This receptor, the one responsible for most of cannabis's psychoactive effects, we now know as the CB1 cannabinoid receptor. It was then successfully cloned, and its distribution, actions, and regulation were thoroughly characterized largely by Howlett's group around 2002.
And wasn't there a second receptor discovered soon after?
Yes, within just a few years, a second cannabinoid receptor, the CB2 receptor, was cloned, this time identified from an immune cell line. So while the psychoactivity you experience from cannabis is overwhelmingly mediated by CB1 receptors, the effects on the immune system, the immunomodulatory effects, are more likely driven by CB2 receptors.
Let's focus on CB1 first then. Where exactly do we find these receptors and what's their primary job?
CB1 receptors are highly expressed in the brain. You find them particularly concentrated on the presynaptic terminals, the sending end of a specific subset of cortical GABAergic interneurons. These are neurons that release the inhibitory neurotransmitter GABA, and they often also co-express another signaling molecule called cholecystokinin, or CCK.
So they're controlling the brakes in the brain, in a way.
That's a good way to think about it in that context, yes. They regulate the release of GABA. But CB1 receptors are also found, albeit at lower levels, on many other nerve terminals throughout the brain. Their role in mediating synaptic plasticity, how connections between neurons change and overall neuronal excitability, is profound. They act both at the preceptic terminal, controlling neurotransmitter release, and sometimes on the neuron's cell body itself.
Does their expression change over time?
Yes,
Yeah.
significantly. CB1 receptor expression changes quite dramatically with age and during development. This likely helps explain why cannabinoids can have different and often more profound or lasting effects on the developing brain compared to the adult brain. And crucially, CB1 receptors aren't just on neurons.
Where else?
They're also expressed in non-neuronal cells like astrocytes, those vital support cells in the brain. And importantly, they're found outside the brain too, in liver cells, hepatocytes, fat cells, adipocytes, skeletal muscle, and various endocrine cells. This really highlights their widespread influence on the body's overall physiology, not just brain function.
Okay, and what about the CB2 receptors? Where are they concentrated?
CB2 receptors have a distinctly different distribution pattern, though equally important. They are highly expressed in immune cells. This includes microglia, which are the brain's own resident immune cells, and also immune cells throughout the body, T cells, monocytes, natural killer cells, neutrophils.
So primarily an immune system receptor.
Predominantly, yes, though they also found at lower levels in some other cell types, including certain neurons, endothelial cells lining blood vessels, parasites which wrap around capillaries, and keratinocytes in the skin. As we mentioned, CB2 receptors are thought to be the main mediators of the immunomodulatory and anti-inflammatory effects of cannabinoids like THC and potentially CBD. There's also growing evidence they could be important targets for reducing drug craving and certain types of pain, suggesting a broader therapeutic potential beyond just immunity.
Now here's where it gets really interesting for me. these aren't just simple on-off switches, are they? You mentioned they're GPCRs. How do they actually signal inside the cell once they're activated?
You're absolutely right. They're far more sophisticated than simple switches. Both CB1 and CB2 belong to this huge family of cell surface receptors called G protein-coupled receptors, GPCRs. You can picture them as intricate antennas on the cell surface. When a signaling molecule, an endocannabinoid or a drug like THC, binds to the outside, it triggers a complex chain reaction, a cascade of events inside the cell.
Okay, what kind of cascade?
Typically, for CB1 and CB2, they couple to inhibitory G proteins, those G proteins, and also to proteins called arrestins. This coupling generally leads to a dampening or modulation of cellular activity, putting the brakes on certain processes.
Right.
However, it's a testament to their versatility that under specific experimental conditions, researchers have also seen them coupled to G's proteins, which actually activate adenylyl cyclase, or to GQ11 proteins, which activate another enzyme called phospholipase C. So they have multiple potential signaling outputs.
But the main pathway is inhibitory.
Yes, their canonical pathways, the most common and well-understood ones when they're coupled to GIDing, involve the inhibition of adenylyl cyclase, which lowers intracellular levels of a key signaling molecule called cyclic AMP. They also inhibit voltage-gated calcium channels, reducing calcium influx into the cell. On the other hand, they lead to the activation of mitogen-activated protein kinases, MAP kinases, which are involved in cell growth and stress responses, and also activate inwardly rectifying potassium channels, known as GRKs, which tend to hyperpolarize the cell, making it less excitable. So they can influence a whole range of cellular processes, from gene expression right down to the fundamental electrical excitability of the membrane.
And you mentioned something really cutting edge earlier, biased agonism. What's that about?
Ah, yes, functional selectivity, or biased agonism. This is a really powerful concept and something professional pharmacologists are intensely focused on. It means that certain ligands, different cannabinoids, for example, don't just turn the receptor on in a generic way. Instead, they can preferentially, or bias, the receptor towards activating specific subsets of those downstream signaling pathways we just talked about, maybe favoring a G protein pathway over an arrestin pathway, or one type of G protein over another.
So two different drugs binding to the same receptor could actually cause different effects inside the cell.
Precisely, because they steer the receptor's signaling down different internal roads. This functional selectivity absolutely has to be considered when we're evaluating the effects of different cannabinoids, especially structurally diverse ones acting at these receptors, maybe particularly CB2. It's a key reason why different cannabinoids can have surprisingly distinct effects, even if they seem to be targeting the same receptor. It adds a whole new layer of nuance.
Okay, so beyond the big two, CB1 and CB2, are there other places in the body where endocannabinoids or cannabis compounds might be having an effect? It feels like the system keeps expanding.
It does seem that way. The picture is definitely more complex. Endocannabinoids and some synthetic ligands aren't strictly limited to interacting with just CB1 and CB2. They can also directly interact with other targets, most notably various ion channels and a class of receptors found inside the cell nucleus called peroxisome proliferator activated receptors, or PPARs.
Ion channels, like directly opening or closing them.
Exactly. ECBs and some synthetic cannabinoids can directly bind to and modulate the activity of several ion channels, including things like 5-HT3 receptors involved in nausea, TRPV1 receptors, pain and heat, GABA receptors, inhibition, and glycine receptors, also inhibition, among others. These are direct interactions separate from their GPCR actions. However, and this is a crucial caveat for professionals critically evaluating the literature, some of these reported direct effects have only been seen in vitro studies, in lab dishes.
And often at high concentrations.
Often, yes, requiring concentrations of endocannabinoids that might be difficult to achieve in vivo in a living organism under normal physiological conditions. So while these interactions are possible, their real-world physiological significance in many cases still needs to be firmly established.
Okay, and what about those PPARs you mentioned? Inside the nucleus.
Right, the peroxisome proliferator activated receptors. Specifically, PPRA and PPRO are known to be activated by endocannabinoids. And this activation might actually contribute significantly to the broader pharmacological effects we see with cannabinoids. PPRs are nuclear hormone receptors. When they get activated, they travel into the cell's nucleus, bind to specific sections of DNA, and directly regulate the expression, the turning on or off, of a whole suite of genes.
What kind of genes?
Genes involved in really fundamental biological processes, regulating metabolism, controlling inflammation, promoting neuroprotection, influencing cellular differentiation. So this suggests that ECBs can exert influence over these core pathways, not just through their surface receptors like CB1 and CB2, but also by going right to the genetic control center via these PPARs. It adds another profound layer of complexity to how they impact our physiology.
Now, let's talk about one of the most fundamental and frankly kind of mind-bending roles of endocannabinoids in the brain, their function as retrograde messengers. That sounds like something out of science fiction. What does it actually mean for synaptoplasticity, which is, how our brains learn and adapt?
It really is one of the most unique and fascinating twists in how neurons communicate. Endocannabinoids are indeed major retrograde messengers in the nervous system, and this role is absolutely fundamental to how our brains learn, adapt, form memories. It mediates several critical forms of synaptic plasticity.
So retrograde means backward.
Exactly. Unlike classical neurotransmitters, which are released from the presynaptic neuron, the sending neuron, and travel forward across the synapse to the postsynaptic neuron, the receiving one.
Right, the usual direction.
ECBs do the opposite. They are synthesized on demand by the postsynaptic neuron, the one receiving the signal. Then, these newly made ECBs travel backward retrogradely across the synaptic cleft to activate presynaptic CB1 receptors located on the axon terminal of the neuron that just sent the signal.
Wow, so the receiving neuron talks back to the sender.
Precisely. By activating those presynaptic CD1 receptors, they suppress further neurotransmitter release from that sending terminal. Imagine a neuron sending a message, and the receiving neuron quickly sends back a signal saying, okay, got it, maybe slow down now, or I need less of that signal for a bit. It's a crucial feedback mechanism for fine-tuning synaptic strength and communication in real time.
How is this retrograde signaling actually triggered?
The induction, whether it leads to short-term or longer-lasting changes, typically requires strong stimulation of that postsynaptic neuron. This usually involves depolarization, a change in its electrical state, which leads to an influx of calcium ions through voltage-sensitive channels, or it can be triggered by the activation of certain GPCRs on the postsynaptic side, specifically those linked to GQ11 proteins, which cause calcium release from internal stores within the cell. This surge in postsynaptic calcium, often working together with GPCR activation, is what switches on the enzyme diacylglycerol lipase alpha, the enzyme that makes 2-AG. Exactly, which then produces 2-AG, the primary ECB involved in most forms of this retrograde signaling, likely due to its higher efficacy and abundance compared to AEA in this context. And you mentioned short-term and longer-lasting effects. Yes, we can categorize this synaptic plasticity based on how long the effect lasts. First, you have the transient forms of plasticity. Two classic examples are DSE, which is depolarization-stimulated suppression of excitation, and DSI, depolarization-stimulated suppression of inhibition. These happen when strong depolarization of the postsynaptic neuron triggers ECB release, which travels back and activates presynaptic CB1s, temporarily reducing neurotransmitter release. DSE occurs if it suppresses an excitatory input, DSI if it suppresses an inhibitory one.
Okay, quick adjustments.
Very quick. Similarly, you have MSE and MSI, metabotropic stimulated suppression of excitation or inhibition. Here, it's the activation of those postsynaptic GPCRs that triggers the 2-AG production and retrograde suppression. These transient forms are remarkably fast. They kick in within a second of the postsynaptic stimulation and typically last for maybe tens of seconds. Perfect for rapid dynamic adjustments in neural circuits, making sure neurons aren't constantly overstimulated.
And the sustained form, LTD.
Right, sustained plasticity, specifically long-term depression, LTD. Unlike those brief DSC-DSI events, sustained lower-frequency activity at excitatory synapses can lead to a more persistent, lasting reduction in synaptic strength mediated by ECBs. The induction of this LTD requires sustained endocannabinoid production, a longer slowdown signal, and this is a really crucial distinction for professionals. Once that LTD is established, its maintenance, keeping that synapse weakened long-term, becomes independent of ongoing endocannabinoid signaling or CB1 receptor activity.
So it locks in a change.
It implies a lasting structural or functional change at the synapse, a genuine form of memory storage at that connection that persists even after the initial ECB signal is gone. The overall impact of all this ECB-mediated plasticity on a network is complex, naturally. It depends on whether the CB1 expressing synapse being modulated is excitatory or inhibitory, how active it is, and the precise spatial relationship between the inputs driving the ECB synthesis and the presynaptic terminals that actually have the CB1 receptors. It's an incredibly sophisticated system, absolutely fundamental to learning, adaptation, and information processing in our brains. It's shaping the very connections between our neurons.
So endocannabinoids are clearly famous for this retrograde messaging, that unique backward talk at the synapse. But do they have other ways of directly influencing how excitable a neuron is, ways that aren't about that feedback loop?
Yes, they absolutely do. While retrograde signaling is arguably their most unique function, endocannabinoids can definitely modify neuronal excitability through several diverse non-retrograde mechanisms as well. The best characterized of these include, as we touched on briefly, direct modulation of various ion channels, activation of G-protein coupled inwardly rectifying potassium channels, GRKs, and also enhancing the activity of a specific channel called the hyperpolarization activated cation channel, or I.
Okay, let's break those down. Direct ion channel modulation.
Right. This means ECBs can physically bind directly to certain ion channels and alter their function, opening, closing, changing how they respond. Channels implicated include, again, 5-HT3, TRPV1, GABA, and glycine receptors, among others. These are distinct from the GPCR-mediated effects. But that same caveat applies here. Some of these reported effects in lab studies require pretty high ECB concentrations.
So we need to be cautious about their physiological relevance in vivo.
Exactly. It's important to critically evaluate whether those concentrations are realistically reached in the living brain under normal circumstances to produce those specific effects. Still an area of active investigation for many of these interactions.
What about those GIRK channels?
The G-protein coupled inwardly rectifying K+ channels, GRKs. This is a well-established non-retrograde effect. Intense neuronal activity can sometimes trigger ECB production to levels high enough to activate CB1 receptors located on the soma, the main cell body of the neuron, not just the axon terminals.
Okay, so acting on the neuron's main body.
Right. And activating these somatic CB1 receptors leads to the opening of GIRK channels. These channels let potassium ions flow out of the neuron, which causes the neuron's membrane potential to become more negative. It hyperpolarizes. This hyperpolarization makes the neuron less likely to fire an action potential, effectively acting as a brake to dampen its overall excitability. It's a key mechanism for preventing runaway neuronal firing.
And the third one, enhancing the IA channel.
Yes, the enhancement of the hyperpolarization activated cation channel, a HIAT. This II channel is really important. It plays a critical role in regulating dendritic excitability, how well the neuron's receiving branches, the dendrites, integrate incoming signals. And because of that, it's central to synaptic plasticity and learning processes.
And what happens when eye activity is enhanced?
Generally, enhancing eye activity tends to impair certain types of learning and memory. And this is particularly relevant because CB1 receptor-mediated enhancement of eye is considered a very plausible mechanism for how THC might impair learning and memory, contributing to some of the cognitive side effects associated with cannabis use.
Is the signaling pathway known?
Yes, it involves a specific cascade inside the cell. It seems to start with activation of a kinase called JNK1, which then activates guanylate cyclase, leading to the production of cyclic GMP. Cyclic GMP then acts on the HCN channels, the channels responsible for the eye current, ultimately enhancing their activity. So you see, the impact of endocannabinoids goes well beyond just tweaking neurotransmitter release. They actively sculpt the fundamental electrical properties and responsiveness of the neurons themselves. Hashtag 3.5. Wide-ranging physiological roles of endocannabinoids in detail.
All right, this is where we really connect the dots. We've got this incredibly intricate system, the messengers, the receptors, the enzymes, the complex signaling actions. So what does it all do? What are the actual wide-ranging physiological roles of the ECS? Where do we truly see its impact across the body and maybe even in our daily lives?
Exactly. This is the crucial so what of our deep dive. The ECS isn't just some obscure molecular pathway. It's deeply involved in regulating so many critical functions, influencing everything from basic cellular processes right up to complex behaviors and our overall state of health. Hashtag 3.5.1. Stress and its resolution. Amygdala. Let's start with perhaps one of the most relevant for many people. Stress and its resolution. It's a very common reason regular cannabis users cite for their use to reduce stress and anxiety. And indeed, the science backs this up. Endogenous ECB signaling itself is a powerful natural mechanism for reducing stress and anxiety.
So our body has its own built-in stress reliever.
In a very real sense, yes. Research in both humans and animals consistently shows that exposure to stress, whether psychological or physical, triggers the mobilization, the release of endocannabinoids, both within the brain and out in the periphery. And glucocorticoids, the body's main stress hormones like cortisol, are actually integral mediators in this ECB response to stress.
And what's the purpose of this ECB release during stress?
Crucially, this rapid release of ECBs following a stressful event seems vital for terminating the stress response, for bringing the system back to baseline. So by enhancing ECB function, either naturally or perhaps pharmacologically, you can actually help restrict or limit the body's physiological and behavioral responses to stress, preventing it from escalating or becoming chronic.
How does it work in the brain?
In the brain, ECBs are particularly active in regions heavily involved in emotional processing, like the amygdala, a key hub for fear and anxiety. Here, ECB signaling acts to gate or dampen stress-induced excitation, effectively restricting the development of anxiety and limiting the release of stress hormones from the HPA axis. Out in the periphery, CB1 receptors located on sympathetic nerve terminals, part of the fight-or-flight system, get activated by ECBs after stress, helping to rein in the autonomic response, like heart rate and blood pressure increases.
What about chronic stress? Does that affect the system?
Yes, and this is a critical point. Chronic stress actually appears to impair ECB function. This loss of the ECB system's ability to effectively buffer and terminate stress responses likely contributes significantly to what scientists call stress-induced allostatic load, the cumulative wear and tear on the body that results from chronic or poorly managed stress.
And didn't you mention that human genetic variant in FAAH again? The one that boosts AEA levels?
Exactly. This ties back beautifully. Remember the P129T variant in the FAAH gene, which leads to reduced FAAH activity and higher baseline AEA signaling. Well, human studies have found associations between carrying this variant and lower levels of trait anxiety, meaning a general predisposition to anxiety, as well as enhanced top-down emotional control, improved ability to inhibit fear responses, and even blunted neural and physiological responses when exposed to stressful situations. It's a powerful demonstration of the system's role.
So that feeling of bliss from anandamide isn't just a fun name. It's literally part of your body's built-in calm-down system, like a natural break on anxiety. That's incredible. Hashtag tag 3.5.2. Reward. Ventral tegmental area VTA.
Absolutely. Now moving on to reward. Cannabis intoxication often produces a state of mild euphoria, and it undeniably possesses addiction potential, although generally considered less severe than substances like opiates. We know it's reinforcing because animals, like rodents, will readily self-administer cannabis vapor and work to get it. These are hallmark laboratory signs of a rewarding substance.
Where in the brain does this happen?
The ability of cannabinoids to enhance the rewarding properties of stimuli seems to be largely mediated by their actions within a crucial brain region called the ventral tegmental area, or VTA. The VTA is a small but vital nucleus packed with dopaminergic neurons, neurons that release dopamine, the brain's primary reward chemical.
And how do cannabinoids affect these dopamine neurons?
It's a neat mechanism. CB1 receptors in the VTA are primarily located on the axon terminals of inhibitory GABA neurons that synapse onto and normally suppress the VTA dopamine neurons. So when cannabinoids activate these presynaptic CB1 receptors on the GABA terminals,
They inhibit the inhibitor.
exactly, they inhibit GABA release. This inhibition of an inhibitory input effectively disinhibits the dopaminergic neurons, allowing them to fire more freely and promoting the release of dopamine within the nucleus accumbens, a major downstream target and key reward center.
And that dopamine release is what feels rewarding.
That dopamine release in the nucleus accumbens is central for encoding the salience of rewarding events, marking them as important, and for motivating us to seek out and engage in rewarding actions. Interestingly, it's not just drugs. Even naturally rewarding stimuli like cocaine in animal models or even voluntary exercise seem to require intact ECB signaling within the VTA to exert their full rewarding effects.
What happens if ECB signaling is disrupted?
Conversely, disrupting ECB signaling can inhibit motivation for rewarding stimuli and reduce engagement in pleasurable activities, potentially leading to a state called anhedonia, the inability to experience pleasure. And linking back to genetics again, that P129T FAAH variant associated with elevated AEA has also been linked in humans to increased neural reactivity of the nucleus accumbens when anticipating or experiencing rewarding cues. And a very important cautionary note for drug development and clinical practice. Pharmacological antagonism of CB1 receptors blocking their action has unfortunately been associated with the development of depression, particularly characterized by anhedonia, hashtag tag 3.5.3, appetite and metabolism, hypothalamus and periphery.
Okay, now for probably the most famous or maybe infamous effect of cannabis, the munchies. How does the ECS actually regulate our appetite and metabolism, not just in the brain, but across the whole body?
Ah, yes, the munchies, a truly prototypical effect and one with deep roots in ECS function. Endocannabinoid signaling is indeed very prominent within the brain's core feeding circuits, especially in the hypothalamus, a major control center for appetite and energy balance. We see that hypothalamic ECB levels naturally fluctuate depending on our nutritional state.
How so?
When you're fasting, ECB levels tend to rise, which helps stimulate appetite and food-seeking behavior. Then, after you eat and feel satiated, these levels decrease. This dynamic regulation allows ECB signaling to rapidly increase or suppress feeding by directly modulating the excitability of specific neuron populations within the arcuate nucleus of the hypothalamus. These include the AGR-PMPY neurons, which are powerful drivers of food intake, and the POMC-MCH4 neurons, which act to inhibit food intake.
Is it linked to hunger hormones too?
Absolutely. The ECS is deeply integrated into the hormonal cascades that regulate feeding. For example, leptin, that powerful appetite-suppressing hormone released from fat tissue, acts rapidly to suppress hypothalamic ECB signaling, contributing to its inhibitory effect on feeding. Conversely, hormones that stimulate hunger, like ghrelin released from the stomach, or stress hormones like glucocorticoids, appear to promote food intake partly by recruiting or enhancing ECB signaling pathways in the hypothalamus. But it's not just the brain. Peripheral ECB signaling also significantly influences both food intake and broader metabolic processes. For instance, stimulating cannabinoid receptors on vagal afferent nerve fibers, those communication lines running from the gut up to the brain and on sympathetic nerve terminals, can also enhance food intake.
Now, you mentioned earlier that excess ECB activity can be problematic metabolically.
Yes, and this is the complex flip side, really crucial for professionals thinking about metabolic health. Chronic or excessive ECB activity, particularly in peripheral organs, can have significant adverse metabolic consequences. For example, persistent activation of hepatic CB1 receptors in the liver is known to promote the development of fatty liver disease, or hepatic steatosis. Similarly, CB1 receptor activation in adipose tissue, in fat cells, can augment adipogenesis, the creation of new fat cells, and increase overall fat accumulation.
Is there evidence for this in humans?
Yes. Consistent with this, elevated levels of endocannabinoids have been observed in rodent models of obesity and, importantly, also in human populations suffering from obesity. This strongly suggests that chronically elevated ECB activity can contribute to the development and maintenance of obesity and related metabolic disorders like type 2 diabetes. And conversely, this is why pharmacological blockade of CB1 receptors using drugs like rimonabant that we discussed produces quite strong anorectic effects, leading to weight loss and preventing many of the metabolic consequences of obesity, like insulin resistance in multiple species, including humans.
Okay, but that brings us back to that really puzzling observation you mentioned, the cannabis paradox. If the system can promote fat storage, why are regular cannabis users often leaner than non-users in large population studies? That seems completely contradictory.
It is a truly curious and important paradox, and one that requires careful consideration of the pharmacology involved. Despite all the evidence suggesting excess endogenous ECB activity can promote obesity, regular cannabis use in humans is generally not associated with higher rates of obesity. In fact, several large-scale studies report that chronic cannabis users tend to have a lower body mass index, BMI, and lower rates of obesity compared to non-users.
What's the leading explanation for this?
One prominent hypothesis centers on the specific nature of THC itself. Remember, THC is a partial agonist at CB1 receptors. It activates them, but not to their full potential, even at high concentrations. In contrast, 2-AG, one of our main endogenous cannabinoids that's often elevated in obesity, is a full agonist, capable of driving maximal receptor activation. So the thinking is that THC, being only a partial agonist, might actually occlude or partially block the receptor, preventing the body's own, more efficacious 2-AG from fully activating it, especially under conditions where endogenous 2-AG levels might be chronically high.
So THC might actually be dampening some of the negative effects of high internal 2-AG.
That's the hypothesis. By acting as a partial agonist, THC could potentially limit some of the negative metabolic consequences that would otherwise be driven by chronically elevated endogenous ECB function. It's a fascinating example of how the subtle pharmacology, partial versus full agonism, can lead to unexpected real-world outcomes and highlights the crucial nuance in understanding how cannabis interacts with our internal system. Hashtag 3.5.4. Pain management, multi-level circuitry. All right, let's move to another profoundly important area, pain management. The use of cannabis for chronic pain and also for chemotherapy-induced nausea represents some of the most common and scientifically well-established therapeutic applications in humans. There's a solid evidence base supporting its efficacy here.
And why is it effective for pain? Where does it act?
Its efficacy likely stems from the incredibly widespread distribution of CB1 receptors throughout multiple levels of the nervous system's pain processing circuitry. We find them at cortical levels in the brain, in key midbrain regions, down in the spinal cord, and even out at peripheral sites where pain signals originate. This suggests cannabinoids tackle pain through a multi-pronged approach.
Can you give some examples?
Sure. Many dorsal root ganglion neurons, the nerve cells that transmit sensory information, including pain, from the body into the spinal cord, synthesize CB1 receptors and then transport them out to their peripheral nerve endings, the afferent fibers. Activating these peripheral CB1 receptors via local ECB signaling can directly suppress the initiation of pain signals right at the source, before the signal even properly enters the central nervous system. Then within the spinal cord itself, ECBs act on CB1 receptors, and possibly sometimes TRPV1 receptors, located within the complex spinal networks that process incoming pain information, further modulating the signal as it ascends towards the brain.
What about higher up in the brain?
Cannabinoids are well known to produce analgesia, or pain relief, by activating CB1 receptors in key brainstem regions, like the periaqueductal gray, PPA, and the rostral ventromedial medulla, RVM. These areas are crucial components of the brain's own descending pain modulatory circuit, a pathway that actively sends signals down to the spinal cord to inhibit incoming pain signals. So cannabinoids can enhance this natural pain control system. Furthermore, ECBs also act in higher-order brain circuits, particularly within the cortical amygdala pathways, involved in processing the emotional or affective component of pain, the suffering, the unpleasantness, rather than just the raw sensory intensity.
Does our body use this system naturally to control pain?
Yes, absolutely. Exposure to noxious or painful stimuli can actually trigger the release of endocannabinoids, both in the periphery near the site of injury and within those distributed supraspinal pain circuits. This suggests ECBs act as endogenous regulators, our body's own attempt to dampen pain initiation and sensitivity. We even see a specific example of this with acute stress. Experiencing acute stress can produce a transient period of analgesia, known as stress-induced analgesia, and this effect is mediated, at least in part, by the local release of ECBs within the PAG. It's like a built-in emergency pain relief mechanism.
And you mentioned an incredible human case earlier that dramatically illustrates this pain-dampening effect, the Scottish woman. Can you remind us about her?
Indeed, it's such a striking and informative case. The Scottish woman was discovered to possess a unique combination of genetic variations. She had not only that P129T FAAH mutation, which slows ADA breakdown, but also a deletion affecting an FAAH pseudogene nearby. The combined effect of these genetic changes resulted in exceptionally robust and sustained high levels of anandamide throughout her body.
And the result?
Clinically, this was associated with a remarkable phenotype. She had what's called congenital pain insensitivity, meaning she felt very little pain, even from significant injuries that would cause other severe pain. And perhaps just remarkably, she also exhibited accelerated wound healing. This unique case provides incredibly powerful, real-world evidence of the profound and multifaceted impact the ECS, particularly AEA signaling, has on both pain perception and tissue repair processes. It really showcases the system's potential when its endogenous signals are significantly amplified. Hashtag 3.5.5. Inflammation, CB2 receptors to AG metabolism. Let's shift gears slightly to inflammation, another critical physiological process where the ECS plays a significant regulatory role, primarily through the CB2 receptors. As we discussed, CB2 receptors are predominantly located on immune cells and in immune tissues, both out in the periphery and within the brain's microglia. Most types of immune cells, T cells, monocytes, natural killer cells, neutrophils, microglia, express CB2 receptors at varying levels.
Then what happens when these CB2 receptors are activated?
Generally, activation of CB2 receptors on these immune cells acts to reduce inflammation. It does this mainly by suppressing the release of pro-inflammatory cytokines, those signaling molecules that drive and sustain inflammation, and also by inhibiting excessive immune cell proliferation and migration to sites of injury or infection.
What about in the brain?
Within the brain, microglial CB2 receptors are particularly interesting. Their expression is often rapidly induced or upregulated in response to inflammation or brain injury. Activating these microglial CB2s helps suppress the release of inflammatory cytokines while simultaneously promoting the release of anti-inflammatory cytokines, contributing to a more balanced and potentially neuroprotective immune response within the brain.
And in the periphery? You mentioned T cells?
Yes. Peripheral T-cell CB2 receptors seem crucial for controlling their migration into tissues, including the central nervous system. They do this partly by reducing the expression of adhesion factors that allow T cells to stick to blood vessel walls and cross into tissues. In fact, Deficits or reduced function of T-cell CB2 receptors have been linked to enhanced infiltration of T-cells into the CNS in conditions like multiple sclerosis, highlighting their importance in regulating autoimmune processes. The downstream signaling pathways, particularly the CB2-mediated activation of MAP kinases, appear integral to promoting these anti-inflammatory effects and perhaps reparative functions as well. While CB1 receptors are found on some immune cells, CB2 receptors really seem to be the primary players mediating most of the direct anti-inflammatory actions of endocannabinoids and potentially phytocannabinoids.
Now, you mentioned something fascinating earlier, that ECBs can regulate inflammation independently of the receptors, involving 2-AG metabolism.
Yes, this is a key insight, particularly for understanding neuroinflammation, and it involves that large pool of 2-AG stored in brain cell membranes. Remember how we said that membrane-associated 2-AG acts as a reservoir for arachidonic acid? Well, under conditions of inflammatory stimuli, the activity of the enzyme MGL rapidly increases.
The enzyme that breaks down 2-AG.
Exactly. This increased MGL activity leads to the rapid breakdown, the catabolism, of this membrane-associated 2-AG. This liberates large amounts of arachidonic acid, which is then swiftly converted by the enzyme COX2 into pro-inflammatory prostaglandins. And critically, the research pinpointed MGL located specifically within astrocytes, the brain's support cells, not neurons, as being responsible for mediating this generation of neuroinflammatory molecules derived from 2-AG breakdown. This highlights a crucial non-receptor-mediated pathway where the endocannabinoid system, specifically 2-AG metabolism, paradoxically contributes to neuroinflammation, showing that even the breakdown products can have significant, and in this case, pro-inflammatory consequences. Hashtag 3.5.6. Sleep, circadian rhythms.
Okay, finally, let's touch upon sleep. The sleep-promoting or prosomnogenic effects of cannabis are very commonly reported and are often a primary reason for continued use, both recreationally and medically. What's the underlying science? How does the ECS influence our sleep and our daily rhythms?
It's a significant and rapidly growing area of research, and the link is becoming clearer. Cannabis use can indeed reduce sleep onset latency, meaning you tend to fall asleep faster. And it can also decrease the number of nighttime awakenings, leading to more consolidated, less fragmented sleep. Subjectively, users often report deeper sleep. Objectively, studies often show it promotes non-rapid eye movement and REM sleep, which includes the deeper slow-wave stages thought to be most restorative, while it often reduces the percentage of time spent in rapid eye movement REM sleep, the stage associated with dreaming. This suggests a direct influence on sleep architecture itself.
Does our internal ECS fluctuate with sleep patterns?
Yes, it does. On an endogenous level, ECB levels, both AEA and 2-AG, naturally fluctuate in the brain and in the circulation in a circadian manner, following our intrinsic daily rhythms. This regular diurnal cycle is known to be disrupted following sleep deprivation, suggesting a direct link between the ECS and our sleep-wake homeostasis, perhaps signaling the body's need for sleep or facilitating recovery during sleep.
What happens if we manipulate ECB levels?
Good question. Experimentally, elevating 2-AG signaling, for instance by using MAGL inhibitors to block its breakdown, has been shown to increase total sleep time, enhance NREM sleep, and suppress REM sleep, mirroring some of the effects of cannabis itself. Conversely, administering a CB1 receptor antagonist, a drug that blocks the receptor, tends to promote wakefulness and arousal and reduces NREM sleep. In humans, there were multiple reports linking the administration of the CB1 antagonist rimonabant to sleep disruption and insomnia.
So the conclusion is,
all this evidence strongly suggests that endogenous ECB signaling contributes significantly to our normative sleep-wake cycles. It seems to be a fundamental part of how our bodies regulate rest, promote sleep consolidation, and perhaps influence the balance between different sleep stages.
so really, whether you're battling stress, trying to get a good night's sleep, dealing with pain, managing inflammation, regulating appetite, or even experiencing reward, your endocannabinoid system is working tirelessly behind the scenes. It's truly a master regulator, deeply integrated into the very fabric of our biology. It's astonishingly widespread. Okay, now that we have this deep appreciation for our body's own endocannabinoid system, let's pivot back to the cannabis plant itself. The plant produces this incredible chemical toolkit, including what we call phytocannabinoids, plant-derived cannabinoids. How are these amazing compounds, like the famous THC and CBD, actually synthesized within the plant's unique biochemical machinery?
It's a really elegant piece of plant biochemistry, and our understanding of these synthetic pathways has grown tremendously in recent years. We don't need to get bogged down in every single enzymatic step, but grasping the overall picture is definitely key for professionals in this field. Both day 9 THC and its non-psychoactive isomer CBD are primarily synthesized in those specialized glandular structures you see on the surface of the plant, called trichomes.
Those little frosty hair-like things.
Exactly, those tiny glistening structures. The highest concentration is typically found on the flowers of the female plant, although lower levels are present elsewhere. Now, the classic phytocannabinoid synthesis pathway starts with some initial biochemical steps that produce two key precursor molecules, geranyl pyrophosphate and oliphthallic acid. These two precursors are then joined together by an enzyme called an aromatic prenyl transferase. This crucial joining reaction forms the initial phytocannabinoid, which is cannabigerolic acid, or CBGA.
CGGI, I've heard that called the mother cannabinoid.
That's right. It's often called the mother of all cannabinoids, or the stem cell cannabinoid, because it serves as the common starting point, the substrate, for the synthesis of most of the other major cannabinoids we know, including the precursors to THC and CBD.
So how do we get from CBGA to THC and CBD?
From CBGA, specific enzymes take over, channeling the pathway in different directions. CBGA primarily serves as the substrate for two key enzymes, E9 tetrahydrocannabinolic acid, THCA synthase, and cannabidiolic acid, CBDA synthase. As their names suggest, THCA synthase converts CBGA into THCA, and CBDA synthase converts CBGA into CBDA.
THCA and CBDA. Those are the acid forms, right? Not the active ones yet.
Precisely. This is a critical point for anyone working with cannabis chemistry or pharmacology. THCA and CBDA themselves have very little, if any, significant psychoactivity or the biological activity we typically associate with THC and CBD. They are the acidic precursors. To become biologically active, they need to undergo a process called decarboxylation.
Which means removing a carboxyl group.
Exactly. It involves the removal of that acidic carboxyl group, COOH. This process is greatly accelerated by heat, which is why smoking, vaping, or cooking cannabis activates the compounds. But it also happens slowly over time with drying, curing, or exposure to light. Decarboxylation transforms THCA into the psychoactive A9-THC and CBDA into the biologically active CBD.
What about other compounds sometimes mentioned, like CBN?
Good point. THCA and THC can also spontaneously oxidize over time, especially with exposure to air and light. This oxidation leads to the formation of cannabinolic acid, CBNA, and then cannabinol, CBN. CBN is often found in aged cannabis material. Importantly, CBN generally lacks the classic psychoactivity of THC and doesn't seem to share the primary biological activities attributed to CBD either, though it might have some mild sedative effects.
So beyond the big three, THC, CBD, CBN, are there other minor cannabinoids in the plant that contribute to its complexity?
Oh, absolutely. The plant synthesizes a whole range of these minor cannabinoids, although usually much smaller quantities than THC or CBD, depending on the specific strain or cultivar and growing conditions. For example, there's cannabichromatic acid, CBCA, which is also formed from CBGA by a different enzyme, CBCA synthase. CBCA then decarboxylates to cannabichromin, CBC. Another one sometimes mentioned is 8-THC.
Delta 8. That's become popular recently.
Yes, 8-THC is a positional isomer of 9-THC, meaning it has the same chemical formula but a slightly different structure. The double bond is in a different position. It's likely formed as a degradation product from THC. Importantly, 8-THC does retain psychoactivity, similar to 9-THC, although many users report it as being somewhat less potent or having a slightly different quality of effect. And then there's a particularly interesting group for professionals to be aware of, the varin cannabinoids.
Varin. What makes them different?
The key distinction lies in their chemical side chain. While THC and CBD both have a 5-carbon pentyl side chain, derived from that oliphthallic acid precursor, the varin cannabinoids possess a shorter 3-carbon propyl side chain. This difference originates early in the synthesis. If a different starting molecule, divernolic acid, is used instead of oliphthallic acid by that initial prenyl transferase enzyme, then you get phytocannabinoids with propyl side chains, like THCV. Exactly. Examples include tetrahydrocannabivarin acid, THCVA, and cannabidivarin acid, CBDVA. These then undergo decarboxylation to form cacocarodyne tetrahydrocannabivarin, THCV, and cannabidivarin, CBDV, respectively. And this structural difference translates into significantly distinct pharmacology. THCV, for instance, is quite different from THC. It acts primarily as a CB1 receptor antagonist or inverse agonist at low doses, meaning it blocks or inhibits the receptor, though it might act as an agonist at higher doses. It's also a low-efficacy CB2 agonist. This unique profile makes THCV interesting for potential applications like appetite suppression or managing diabetes, quite different from THC's effects.
This complex chemistry brings up an important practical point. What exactly does it mean when we talk about a high CBD or high THC strain? It seems it's all about which enzyme, THC asynthase or CBD asynthase, is more active in that particular plant, right? And that has huge implications, not just for the effects, but legally too, especially here in the US.
Precisely. In most cannabis cultivars, the levels and activity of THC synthase and CBD synthase tend to vary reciprocally. It's like they're competing for the same CBGA precursor.
Yeah.
So plants that have been selectively bred over generations to have very high THCA synthase activity will produce large amounts of THCA, which becomes THC upon heating, and consequently very low levels of CBD.
The classic high THC marijuana strains.
Exactly. Conversely, plants specifically bred for high CBD synthase activity, often referred to as hemp cultivars, will produce high levels of CBDA, which becomes CBD, and typically very little THCA. Through intensive selective breeding, cultivators can now achieve astonishingly high total THC content in dried flower, sometimes exceeding 30% by weight. This potency has significant implications for users.
And the legal side.
The legal side hinges on that THC content. It's also crucial to remember that the enzyme specificities aren't absolutely perfect. Even plants primarily bred to produce high CBD can still synthesize detectable, albeit usually low, levels of THC. This is a critical point for legal frameworks. In the United States, for example, cannabis plants containing greater than 0.3% total THC by dry weight are legally classified as marijuana, a THC-producing cannabis, and fall under DEA jurisdiction, distinct from hemp, which must be below that threshold.
What about that idea that CBD can turn into THC in the stomach? Is that a real concern?
That's an interesting chemical point. It is chemically possible for CBD to convert into THC under mildly acidic conditions, like those potentially found in the stomach. However, whether this conversion actually occurs to a physiologically significant extent in the human body under normal conditions, enough to produce noticeable psychoactive effects from consuming CBD, is not unequivocally established. It remains a point of scientific debate and requires more definitive research. And finally, we shouldn't forget that cannabis plants produce more than just cannabinoids. They also synthesize a rich array of other compounds, notably terpenes and flavonoids.
Terpenes, those are responsible for the smell, right?
Yes. Terpenes are volatile organic compounds that give different cannabis cultivars their distinctive aromas, citrusy, piney, earthy, floral notes. They're built from isoprene units and come in a huge variety of structures. While they undoubtedly contribute to the sensory experience, the exact extent to which these terpenes actually contribute to or modify the psychoactivity or therapeutic benefits of cannabis, the so-called entourage effect, is still an area of very active research and considerable debate. We don't fully understand those interactions yet. Flavonoids are another class of compounds contributing to plant color and likely offering antioxidant or anti-inflammatory properties, but their specific interactions with the ECS are also still being explored.
Okay, so we've covered the plant's own complex chemistry. But then there's this whole other category, synthetic cannabinoids created entirely in labs. What's the story behind these? It sounds quite different from the natural compounds.
It's a very different story and really a stark dual narrative. On one hand, there's been considerable legitimate scientific effort in medicinal chemistry to synthesize novel synthetic cannabinoids, specifically targeting CB1 receptors, often with the hope of developing new therapeutic applications. Think potent, non-addictive pain relievers, for example. This research has generated a rich library of cannabinoid agonists belonging to diverse chemical structures, offering potential avenues for drug discovery with perhaps more targeted or refined effects than THC.
That sounds promising, but there's a darker side.
There is indeed a much darker, more dangerous side that emerged, particularly in the illicit recreational drug market. The motivation here was largely driven by drug testing. Standard drug tests screen for THC and its metabolites, and these metabolites can linger in the body for a long time, weeks even, leading to positive tests long after use.
So people wanted something that wouldn't show up.
Exactly. Individuals subject to drug testing, perhaps for employment or legal reasons, sought alternatives that would provide similar effects but evade detection. Because the chemical structures of many of these early synthetic cannabinoids were substantially different from THC, they could initially slip under the radar of common urine drug screens. This loophole fueled the rapid and widespread recreational use of a vast array of these compounds, often sold under misleading brand names like Spice or K2, frequently marketed as herbal incense or potpourri and labeled not for human consumption.
But these spice products turned out to be incredibly dangerous, didn't they?
Extremely dangerous. This is a critical point for professionals and the public alike. There are several reasons why. First, almost all synthetic cannabinoids found in these recreational products are high-efficacy CB1 agonists. Unlike THC, which is a partial agonist, these synthetics often fully and maximally activate the CB1 receptor, sometimes even more strongly than our own endocannabinoids. This can lead to profoundly exaggerated, overwhelming, and unpredictable physiological and psychological effects.
What else makes them risky?
Second, their off-target activities are often completely unknown or uncharacterized. Because they were synthesized illicitly, often based on old scientific literature without further safety testing, nobody knows what other receptors or enzymes in the body they might be interacting with. This can lead to a completely unpredictable range of additional effects beyond just CB1 activation, contributing to severe and unexpected psychoactivity, including acute psychosis, paranoia, agitation, seizures, and even cardiovascular events like heart attacks or strokes.
And the manufacturing process itself is risky.
Absolutely. Third, and crucially, these synthetic cannabinoids are typically prepared in clandestine, unregulated labs with little to no quality control. The final product sprayed onto inert plant material can have wildly inconsistent dosages hotspots with dangerously high concentrations. It might be contaminated with residual solvents, synthetic byproducts, or even intentionally adulterated with other dangerous substances. This complete lack of quality control adds another layer of extreme unpredictability and danger.
It's a really stark reminder, isn't it, that synthetic doesn't automatically mean safer or more predictable. In this realm, often quite the opposite, the lack of regulation and testing turned potential promise into real peril. Hashtag tag 4.3, interactions between ECBs, THC, and synthetic cannabinoids.
That's a critical takeaway, and it leads us nicely into understanding the sometimes complex interactions between different types of cannabinoids, our own endocannabinoids, THC from the plant, and these synthetic compounds. The key factor driving these interactions is often their varying intrinsic efficacies at the CB1 receptor. Remember, 2-AG is highly efficacious, a full agonist. AEA is a low-efficacy agonist. THC is also potent, but considered a low-efficacy or partial agonist. And those recreational spice compounds are typically highly efficacious full agonists.
So how does this play out? For example, how might THC interact with our internal 2-AG?
That's a great question with nuanced answers. Because THC is a potent but low-efficacy partial agonist, and 2-AG is a highly efficacious full agonist, their interaction depends on the context. Under certain conditions, maybe where CD1 receptor density is low, or the downstream signaling machinery is limited in a particular synapse or tissue, THC might actually function as an antagonist towards endogenous 2-AG signaling.
Oh.
Because THC would bind strongly to the receptor, has high affinity, but only activate it partially. In doing so, it would block the more efficacious 2-AG from binding and exerting its full effect. It's like putting a dimmer switch in place of a full-off switch. However, under different circumstances, perhaps where CB1 receptor density is very high and the signaling capacity is ample, both THC and 2-AG might produce similar maximal cellular effects, because even THC's partial activation might be enough to saturate the downstream response in that specific context.
Can we see evidence of this complexity in humans?
Possibly. It might help explain some intriguing clinical observations. For instance, some human behavioral studies using the CB1 antagonist, rimonabant, found that even very high doses of the antagonist only weakly block the subjective effects of THC, like the high. But the same antagonist strongly attenuated THC-induced tachycardia, the increased heart rate. This suggests that different effects of THC might be mediated by different levels of CB1 activation or occur at different synapses where these efficacy dynamics play out differently. So whether THC ultimately antagonizes, mimics, or even augments endogenous ECB action likely varies significantly across different brain regions and physiological systems. It's not a simple one-size-fits-all interaction.
What about the spice compounds interacting with our internal system?
That interaction is likely much more straightforward and concerning. Since spice compounds are typically highly efficacious full agonists, they forcefully and indiscriminately activate CB1 receptors, likely overpowering the more subtle signaling of our endogenous endocannabinoids. They would almost certainly counter the nuanced signaling of AEA, for example, given AEA's low efficacy. The potent spice compounds would outcompete AEA and drive the receptor to maximal activation, potentially disrupting the delicate homeostatic balance maintained by the ECS.
And this high efficacy contributes to their danger.
Absolutely. The very high efficacy of these spice compounds, combined with their potential to hit unknown off-targets and the frequent presence of dangerous adulterants in illicit formulations, provides a strong pharmacological explanation for their propensity to produce significantly more frequent and severe adverse effects than THC, including those dangerous outcomes like psychosis, seizures, kidney damage, and cardiovascular emergencies. It really highlights for professionals how crucial understanding concepts like intrinsic efficacy is even within the same receptor system. These subtle differences in how a compound binds and activates can completely change its physiological impact, making the pharmacology incredibly nuanced.
Okay, let's move from the complexities of illicit synthetics to medications that have gone through regulatory approval. Let's start with dronabinol. This is basically chemically synthesized THC, the main psychoactive compound from cannabis, but made in a lab and formulated as a medicine. What's its story, and how is it actually used clinically?
Right, dronabinol is essentially pharmaceutical-grade synthetic delta-9 THC. In the U.S., it's available primarily as THC dissolved in sesame oil within a gelatin capsule and also as an oral solution. Its mechanism of action is pretty straightforward. Just like THC from the plant, dronabinol acts as a low-efficacy or partial agonist at both CB1 and CB2 cannabinoid receptors. Now, the ADME absorption, distribution, metabolism, and excretion is really crucial for understanding how dronabinol works clinically, especially because it's taken orally. When you take it by mouth, it's actually absorbed quite well from the gastrointestinal tract. However, it then undergoes significant first-pass metabolism in the liver.
Meaning the liver breaks down a lot of it before it gets into the main circulation.
Exactly. A large portion of the absorbed drug is metabolized by the liver on its first pass-through before it reaches the rest of the body. The result is a relatively low oral bioavailability, typically only about 10% to 20% of the ingested dose actually makes it into the systemic circulation as unchanged THC.
When do blood levels peak after taking it?
Peak plasma THC concentrations usually occur about 1 to 3 hours after taking the capsule. Interestingly, taking dronabinol with food, especially fatty food, can slow down the time it takes to reach that peak concentration. But it generally increases the overall exposure, the total amount of drug the body sees over time, although maybe not the absolute peak level itself. This suggests patients should probably aim for consistency, either always take it with food or always on an empty stomach to get predictable effects. The metabolism of THC, including dronabinol, is a really critical piece of the puzzle. THC is primarily metabolized through successive oxidation, mainly at the carbon-11 position. This first step produces 11-hydroxytetrahydrocannabinol, or 11-OH-THC. This metabolite is then further oxidized to an inactive compound called 11-norcarboxy-THC, which is what drug tests often look for.
Is that 11-OH-THC metabolite important?
Extremely important. This is absolutely crucial for professionals to grasp. 11-OH-THC retains high activity at CB1 cannabinoid receptors. It's a potent psychoactive metabolite. In fact, after oral consumption of dronabinol, the plasma levels of 11-OH-THC can actually be higher than the levels of the parent THC compound. This means that most of the biological and psychoactive effects experienced after taking oral dronabinol are likely due to this active metabolite, 11-OH-THC, rather than the THC itself.
That's a key difference from smoking or vaping then.
A huge difference. When THC is inhaled, it bypasses that extensive first-pass metabolism in the liver. You get much higher bioavailability, maybe 60–70%, much higher peak blood levels of THC itself occurring very rapidly, within minutes, a faster decline, and a shorter duration of intoxication, typically 90 minutes to maybe 4 hours. Oral dronabinol gives you lower blood levels of THC, a much higher ratio of 11-OH-THC, a slower peak, 1 to 3 hours, a much longer duration of effects, potentially lasting 8 to 12 hours. Very different pharmacokinetic profiles lead to very different experiences.
How is it eliminated from the body?
Excretion of dronabinol and its metabolites is mainly via the bile into the feces, with the smaller amount appearing in the urine, and the metabolism is quite prolonged. Those inactive metabolites can be detected in urine and feces for more than 5 weeks after just a single dose, which has obvious implications for drug testing.
What are the approved uses for dronabinol in the U.S.?
It's approved for 2 specific therapeutic uses. First, for treating severe nausea and vomiting associated with cancer chemotherapy, specifically in patients who haven't responded adequately to standard antiemetic treatments. Second, it's approved for treating anorexia, loss of appetite, associated with significant weight loss in patients with AIDS, aiming to stimulate appetite and help them regain weight.
What about side effects and potential drug interactions?
The most common adverse effects involve the central nervous system, CNS, and the cardiovascular system. CNS effects can include things like dizziness, drowsiness, fatigue, problems with concentration or memory, and sometimes euphoria or dysphoria. Importantly, it can also potentially exacerbate pre-existing psychiatric conditions like mania, depression, or schizophrenia, so caution is needed in those patients. Cardiovascular effects typically include tachycardia, increased heart rate, and sometimes hypertension, changes in blood pressure. Occasionally, patients might paradoxically experience increased nausea, vomiting, or abdominal pain, which could, in rare cases, be a sign of cannabinoid hyperemesis syndrome, especially with chronic high-dose use.
How are these managed?
Usually by holding or reducing the dose, or sometimes by administering the dose before sleep if CNS effects like dizziness are problematic during the day. From a drug interaction perspective, droneobanol's metabolism relies mainly on the CYP enzyme CYP2C9, which seems to play a larger role, and CYP3A4. This means there's a definite potential for interactions with other drugs that might induce or inhibit these specific enzymes. Taking droneobanol with a strong CYP inhibitor could increase THC-11-OH-THC levels and side effects, while taking it with an inducer could lower levels and reduce efficacy. Conversely, high doses of THC might potentially impair the metabolism of other drugs that also rely on these CYPs.
Is there genetic variability in how people handle it?
Yes, particularly with CYP2C9, which is known to be highly polymorphic, meaning there are common genetic variations that affect its activity. Individuals who are homozygous for the CYP2C93 variant, for example, are considered poor metabolizers. They might break down THC much more slowly and could be unusually susceptible to adverse effects or require lower doses. Droneobanol isn't approved for pediatric use, and while there's been some research interest in using it for agitation and dementia, it's not currently approved for that indication either.
How is it typically dosed in practice?
For clinically use in chemotherapy-induced nausea and vomiting, CINV, it's often dosed based on body surface area, typically starting at 5 mg, given 1 to 3 hours before chemotherapy, and then repeated every 2 to 4 hours after chemotherapy for a total of 4 to 6 doses per day. The dose can be increased cautiously in increments of 2.5 mg up to a maximum of 15 mg per dose if needed, but side effects increase significantly at higher doses, so careful monitoring is essential. For AIDS-related anorexia, the usual starting dose is 2.5 milligrams twice daily, typically taken before lunch and again before dinner or sleep. If that's well tolerated but not effective enough after a few days, the dose can be slowly increased, potentially up to 10 milligrams twice daily, though often lower doses are sufficient. Adjustments might be needed based on side effects, like reducing the daytime dose if it causes too much drowsiness. #TAG 5.2. The Bixable, standardized cannabis extract THCBD.
Okay, let's move on to nabiximols. Now, this is different from droneoban because it's a standardized extract from the cannabis plant itself, usually containing roughly equal amounts of THC and CBD plus other plant components. How does this formulation work, and where is it used?
Right, nabiximols is quite distinct. It's a carefully prepared and standardized extract derived directly from specific cannabis cultivars, ensuring consistent ratios of its main components. It contains approximately equal amounts of THC and CBD, but importantly, it also includes smaller quantities of other minor cannabinoids, various terpenes, and triglycerides that are naturally present in the source plant material. It's formulated as an ethanol-containing, peppermint-flavored metered dose oral mucosal spray.
Or mucosal, meaning sprayed into the mouth.
Exactly. It's sprayed typically under the tongue or onto the inside of the cheek, allowing for absorption primarily through the lining of the mouth, the buccal mucosa, although some is inevitably swallowed and absorbed through the gut too. It's important to note upfront that nabiximols is not currently FDA approved for use in the United States, but it is approved and widely available in many other countries, including Canada, the UK, and several European nations.
What's its mechanism of action considered to be?
Its mechanism of action is thought to be primarily driven by its two main active ingredients. You have THC acting as that low-efficacy partial agonist at both CB1 and CB2 receptors, and then you have CBD, which, as we've discussed, seems to have a more complex profile, potentially interacting with multiple targets beyond the cannabinoid receptors, like serotonin 5-HT1A receptors or adenosine signaling pathways, possibly contributing anti-inflammatory or anxiolytic effects. The specific role of the other minor constituents, the other cannabinoids, the terpenes, and its overall therapeutic efficacy, is still an area of active research and contributes to that ongoing discussion about the entourage effect. Is the whole extract more effective than THC or CBD alone? That's the key question. There's also the potential for pharmacokinetic interactions between THC and CBD within the product, as they share some overlapping CYP metabolic pathways, which could influence their respective blood levels and effects.
How well is it absorbed, and is it consistent?
The ADME for nabiximols is characterized by notable variability between patients. This is partly due to that mixed absorption route, some through the mouth lining, some swallowed. Peak plasma concentrations of THC typically occur about 1 hour, or maybe a bit longer, after spraying into the mouth. However, both the peak levels achieved and the overall exposure area under the curve, for both THC and CBD can vary quite substantially between different individuals receiving the same number of sprays, sometimes up to 10-fold differences in peak levels, and 3 to 4-fold differences in overall exposure have been reported. This high inter-individual variability is definitely a key factor to consider in clinical practice.
Does food affect its absorption?
Yes, significantly. Consuming food, particularly fatty food, around the time of administration can markedly increase peak plasma levels and overall exposure for both THC, roughly doubling them, and CBD, increasing them even more, perhaps fourfold. This underscores the importance for patients to be consistent in how they use the spray relative to meals to maintain relatively stable drug levels and achieve predictable effects day to day. Compared to inhaling cannabis, the peak levels of THC and CBD reached with the oromucosal spray are much lower and achieved more slowly, leading to a more gradual onset and potentially more sustained effect profile.
What about metabolism and drug interactions?
The metabolism of nabiximols is dominated by the metabolism of its main components, THC and CBD, primarily via those CYP enzymes we've discussed, like CYP2C9, CYP304, CYP2C19. Importantly, nabiximols itself, likely due to the CBD component, has been shown to inhibit the enzyme CYP3A4 in a time-dependent manner, even at clinically relevant doses. This creates a significant potential for nabiximols to increase the blood levels of other drugs that are also metabolized by CYP3A4. Conversely, strong CYP3A4 inhibitors, like the antifungal ketoconazole, can substantially increase exposure to THC and CBD from nabiximols, while strong CYP3A4 inducers, like the antibiotic rifampin or the supplement St. John's wort, can significantly decrease THC and CBD exposure, potentially reducing efficacy. So careful review of co-administered medications is essential to avoid adverse interactions or loss of effect.
What is nabiximols primarily approved for?
Its main therapeutic use, for which it's approved in several countries, is as an add-on treatment for moderate to severe spasticity associated with multiple sclerosis, MS, specifically in patients who haven't responded adequately to other antispasticity medications. It's also sometimes used for neuropathic pain associated with MS. There has been considerable interest in using nabiximols for other types of chronic pain, particularly neuropathic pain, but the clinical trial data supporting these broader indications have been somewhat mixed and less conclusive than for MS spasticity. It is not approved for pediatric use.
What are the typical side effects?
Common adverse effects reported with nabiximols generally include dizziness, drowsiness, fatigue, and sometimes issues with memory or concentration. These are largely attributable to the THC component acting on CB1 receptors in the brain. Usually, these side effects are reported as mild to moderate in intensity, and importantly, they often tend to diminish with continued use over days or weeks, suggesting that patients develop some degree of tolerance to these effects. Management typically involves temporarily holding the dose or reducing the number of sprays per day if side effects become bothersome.
How is it typically used in practice? How do patients find the right dose?
In clinical use, each 100 microliter spray delivers a precise dose, 2.7 milligrams of THC and 2.5 milligrams of CBD. Because of the potential for side effects, especially initially, therapy is usually initiated using a careful titration schedule. Patients typically start with a single spray in the evening for the first couple of days, then gradually increase the number of sprays, often adding another evening spray, then adding morning doses, spreading the total number of sprays throughout the day based on their individual response and tolerability. The goal is to find the lowest effective dose that provides adequate symptom relief, e.g., reduced spasticity, without causing unacceptable side effects. The maximum recommended dose is typically around 12 sprays per day. The titration process allows patients to slowly acclimatize and find their optimal dose. If a patient doesn't experience significant improvement in their spasticity symptoms after about 4 weeks of consistent use and titration, it's generally recommended to discontinue treatment, as further benefit is considered unlikely. Hashtag 5.3, cannabidiol, CBD, oral solution.
Okay, now let's talk about cannabidiol, or CBD. This compound has absolutely exploded in popularity worldwide. You see it advertised everywhere in seemingly everything from oils and gummies to coffee and skincare. But what does the rigorous pharmaceutical science say about its approved uses and how it actually works? We're talking specifically about the FDA-approved oral solution of cannabis-derived CBD here, right? Not the over-the-counter stuff.
That's a critical distinction, yes. We're focusing here on the pharmaceutical-grade CBD oral solution, which is derived from cannabis, but manufactured under strict quality controls, and is FDA approved for treating certain severe medication-resistant seizure disorders in children and adults. This is very different from the vast, largely unregulated market of over-the-counter OTC CBD products. Now, what's particularly interesting, and honestly still somewhat debated within the scientific community, is the precise mechanism of action by which this pharmaceutical CBD actually reduces seizures. It doesn't appear to primarily work through directly engaging the classical cannabinoid receptors, CB1 or CB2, in the same way THC does. Its anticonvulsant mechanism likely involves other targets or pathways that are still being fully elucidated. However, some of the CBD's other reported pharmacological actions have somewhat clearer potential mechanisms. For instance, its anxiolytic or anxiety-reducing effects observed in some studies seem to involve activation of serotonin 5-HT1A receptors. And its anti-inflammatory actions might be mediated, at least in part, by potentiating the signaling of adenosine, an important endogenous molecule involved in energy balance and neurotransmission.
How does the body handle this oral CBD solution? What's its ADME profile like?
Looking at ADME for the oral CBD solution, peak plasma concentrations are typically reached relatively slowly, around 3 to 5 hours after taking it. A really key point for professionals is its oral bioavailability. Generally, it's quite low and highly variable when taken on an empty stomach. Only a small and inconsistent fraction of the dose might make it into the bloodstream. However, this bioavailability is dramatically enhanced, potentially anywhere from 3-fold to even 15-fold higher when CBD is taken concomitantly with fatty food.
Wow, that's a huge difference. So taking it with a meal containing fat is crucial for absorption.
It appears to be extremely important for achieving higher and potentially more consistent blood levels. This food effect is a major consideration for dosing and ensuring reliable efficacy. CBD's elimination half-life is quite long and also variable, ranging perhaps from 14 to over 30 hours, and it tends to increase with chronic dosing, meaning it can accumulate in the body over time with repeated administration.
How is it metabolized?
CBD is primarily metabolized in the liver, mainly by hydroxylation at the C7 position, an action carried out chiefly by the enzyme CYP2C19. This C7 hydroxy metabolite is then further oxidized, mainly by CYP3A4, to a C7 carboxylic acid. Interestingly, that initial metabolite, 7-hydroxycannabidiol, 7-OH-CBD, appears to be analogous to 11-OH-THC in that it seems to retain anti-epileptic activity itself. So, like THC, CBD might have an active metabolite contributing to its effects. CBD metabolites also undergo phase 2 glucuronidation before being eliminated, mostly in the feces, with only small amounts excreted via urine.
What specific conditions is this pharmaceutical CBD approved for?
For therapeutic uses, the FDA-approved CBD oral solution is specifically indicated for treating medication-resistant seizures associated with 3 rare but severe epilepsy syndromes: Dravet syndrome, Lennox-Gastaut syndrome, LGS, and seizures associated with tuberous sclerosis complex, TSC. Clinical trials are also ongoing to evaluate its efficacy in other difficult-to-treat epilepsies.
What about its potential for other conditions like anxiety or psychosis that get talked about a lot?
Beyond the approved seizure indications, the evidence is still developing or mixed. While some experimental studies have shown that acute high doses of CBD can reduce measures of social anxiety in specific controlled settings, its broad efficacy for treating diagnosed anxiety disorders is not yet definitively established by large-scale trials. There's also been significant interest in CBD as a potential antipsychotic agent. However, clinical trial results here have been inconsistent. Some smaller studies suggested benefits, for instance, improving cognitive symptoms or positive symptoms, like hallucinations, in schizophrenia, while others found no significant effect compared to placebo. So its role in psychosis needs much more research and clarification.
Any potential in addiction?
There is some promising preliminary evidence. One notable study found that giving 400 or 800 milligrams of CBD per day for just 3 days significantly reduced craving and anxiety triggered by drug-related cues in individuals who were abstinent from heroin use. This suggests CBD might have potential as a therapeutic agent for certain aspects of substance use disorders, but again, larger trials are needed.
Given the high doses used for epilepsy, what are the main concerns regarding side effects and drug interactions?
This is really important, especially concerning adverse effects and drug interactions. The most common adverse effects reported in the epilepsy trials at those high therapeutic doses, often 10-20 milliliters a day, include somnolence, drowsiness, fatigue or lethargy, decreased appetite, and diarrhea. But a particularly significant concern is the potential for elevated hepatic transaminases, which are markers of liver stress or injury.
Liver issues, how common is that?
These liver enzyme elevations appear to be dose-related. In the clinical trials, elevations in ALT, alanine aminotransferase, greater than 3 times the upper limit of normal were observed in over 10% of patients taking CBD at doses of 10 to 25 milliliters per day. While about a third of these elevations resolved, even with continued CBD treatment, it's a serious potential side effect. The risk seems to be almost doubled in patients who are also taking another common anti-epileptic drug, valproate, with or without a third drug, clobazam. Therefore, regular monitoring of serum transaminases and bilirubin levels is mandatory for patients receiving pharmaceutical CBD, especially when starting therapy or after increasing the dose.
And drug interactions must be a big concern too.
Absolutely. Given the high doses of CBD used, the fact that these patients are often on multiple other antiepileptic drugs, which themselves can affect CYP enzymes, and CBD's own known interactions with several CYP enzymes, it inhibits CYPO, 2C19, and CYP3A4, for example. CBD has a high potential for significant drug interactions.
Any specific ones to watch out for?
The best documented and perhaps most clinically significant interaction is with the antiepileptic drug clobazam. Plasma levels of clobazam itself, and especially its active metabolite, norclobazam, often increase dramatically when CBD therapy is initiated. This is consistent with CBD inhibiting CYP3A4 and CYP2C19, the enzymes responsible for metabolizing clobazam and norclobazam. Therefore, monitoring clobazam levels and potentially adjusting its dose is often necessary when starting CBD. Interestingly, while co-administering Valpro with CBD doesn't seem to increase Valpro levels, it does significantly increase the risk of that liver transaminase elevation, reinforcing the need for careful liver monitoring in patients on both drugs.
Is it approved for children?
Yes, the FDA-approved formulation is approved for treating those specific seizure disorders in patients aged 1 year and older, making it a valuable option for these often very difficult-to-treat childhood epilepsies.
How is it dosed clinically?
In clinical use for seizures associated with Lennox-Gastaut or Dravet syndrome, CBD is typically initiated at a dose of 2.5 milligrams twice daily. This is then usually increased after 1 week to a maintenance dose of 5 milligrams twice daily. If further seizure control is needed and the drug is tolerated, the dose can be further increased in weekly increments up to a maximum maintenance dose of 10 milligrams twice daily. For seizures associated with TSC, the starting dose is also 2.5 milligrams twice daily, but the titration typically goes up to a higher maximum maintenance dose of 12.5 milligrams twice daily, again increased gradually based on response and tolerability. These are very high doses compared to OTC products.
Okay, this brings us right back to that crucial point you made earlier, the OTC CBD conundrum. If the science shows these really high doses, hundreds or even over 1,000 milligrams a day, are needed for proven efficacy in epilepsy, what are people actually experiencing when they take those typical low-dose CBD oils or gummies, maybe 5, 10, 25 milligrams a day, that you find everywhere? It feels like there's a huge disconnect. Where does the line between real pharmacology and maybe psychology get blurry here?
This is arguably one of the most important distinctions for listeners to grasp, truly separating the professional, evidence-based understanding from the widespread marketing hype and anecdotal reports surrounding CBD. The dose discrepancy is enormous. As you said, the effective doses in rigorous clinical trials for epilepsy are consistently very high, starting around 200 milligrams per day and going up to 1,600 milligrams per day in some studies. In stark contrast, the doses people typically consume from readily available non-prescription over-the-counter CBD preparations are drastically lower, usually in that 5 to 25 milligram per day range, sometimes even less.
So pharmacologically, can those low doses even do anything?
Given the generally poor and variable oral bioavailability of CBD, especially when taken without fat, and the lack of any clearly identified high-affinity pharmacological targets for CBD at the extremely low nanomolar blood concentrations that would likely be achieved by these tiny OTC doses, well, it becomes highly questionable whether these low doses are producing any direct, measurable biological or pharmacological impact. There just isn't a known mechanism by which such low levels would be expected to exert significant physiological effects based on current scientific understanding.
So what explains the benefits many people report feeling from low-dose OTC CBD?
There's a very strong reason to believe that expectancy bias and the placebo effect are playing a powerful, perhaps dominant role here. If someone believes a product will help them feel less anxious, sleep better, or have less pain, that belief itself can trigger real physiological changes and perceived improvements. The placebo effect is a genuine neurobiological phenomenon, but it's driven by expectation and context, not by the specific pharmacological action of the substance at that low dose. So for individuals claiming significant benefits from typical low-dose OTC CBD products, it's highly probable that these perceived effects are primarily attributable to these powerful placebo mechanisms rather than a direct drug effect of the CBD molecule itself at those concentrations.
So what this all means for someone just curious about trying CBD is that there's a very significant difference between the anecdotal experiences often reported with low-dose OTC products and the scientifically proven effects seen only with much, much higher doses of pharmaceutical-grade CBD under medical supervision for specific conditions. The marketing often blurs this line, but the science is becoming clearer.
Precisely. It underscores the importance of relying on rigorous scientific evidence and being critical of marketing claims, especially when dealing with unregulated products and doses far below those shown to be effective in clinical trials. #5.4 Nabilone, synthetic THC analog.
All right, one last medication in this category to cover, nabilone. What is this drug, and how does it compare to dronabinol or THC itself?
Nabilone is another synthetic cannabinoid specifically designed as an analog of THC. It's structurally distinct from delta-9 THC, but shares similar pharmacological effects. It was previously FDA approved and marketed in the U.S. as capsules. However, it's important to note that nabilone was actually discontinued in the U.S. market back in 2019.
Why was it discontinued?
The discontinuation was reportedly due to rare but serious hypersensitivity reactions being observed. However, nabilone is still available and used therapeutically in other countries, like Canada, the UK, and Mexico.
How does its mechanism compare to THC?
Its mechanism of action is very similar to THC. It acts as a low-efficacy or partial agonist at both CB1 and CB2 cannabinoid receptors. Interestingly, some studies suggest nabilone might be somewhat more efficacious than THC itself at activating both of these receptors, meaning it might produce a slightly stronger maximal response at the receptor level compared to THC, though still acting generally as a partial agonist.
What about its ADME characteristics?
For its ADME, nabilone is taken orally in capsule form. It boasts very high absorption from the gastrointestinal tract, estimated at around 96%. Peak plasma concentrations are typically reached about 2 hours after ingestion. Like THC, nabilone undergoes significant first-pass metabolism in the liver, but its pharmacokinetics appear to be relatively linear over the typical clinical dose range of 1 to 4 milligram, meaning doubling the dose roughly doubles the exposure. The full details of its distribution and metabolism haven't been as comprehensively studied as THC's. Metabolism is primarily hepatic in the liver and likely involves several CYP enzymes, similar to THC. Key metabolic steps include hydroxylation on its dimethylheptyl side chain and the formation of carboxylic acid analogs. There's also evidence that the ketone group on carbon 9 can be reduced to form various isomeric alcohol metabolites. These metabolites are primarily excreted via the feces.
What are its main therapeutic uses in countries where it's still available?
Its primary indication, similar to dronabinol, is for the management of chemotherapy-induced nausea and vomiting, CINV, specifically in patients who haven't responded adequately to conventional antiemetic therapies. It has also seen various off-label uses in clinical practice, including for managing nightmares associated with PTSD, for certain types of chronic neuropathic pain, sometimes for managing cannabis withdrawal symptoms in hospitalized settings, and occasionally for agitation in patients with early dementia.
What about its side effect profile and potential interactions?
Regarding adverse effects and drug interactions, nabilone's side effect profile largely mirrors that of dronabinol and THC, primarily involving the CNS and cardiovascular system. CNS effects can include drowsiness, dizziness, vertigo, euphoria, dysphoria, dry mouth, and impaired concentration or coordination. Some patients might experience increased anxiety, nervousness, or even panic. In rare cases, particularly at higher doses, hallucinations or delusional thinking have been reported. Sedation and cognitive impairment tend to become more prominent at higher doses, for example, 5 milligrams or more. Cardiovascular effects can include mild tachycardia, increased heart rate, and postural hypotension, a drop in blood pressure upon standing, especially doses of 2.5 to 5 milligrams or higher. These effects, particularly postural hypotension, might occur more frequently in elderly patients.
Any known drug interactions?
While nabilone's metabolism isn't fully defined, it likely involves the same CYP enzymes as THC, like CYP2C9 and CYP3A4. Therefore, there's a theoretical potential for drug interactions with inhibitors or inducers of these enzymes, although no major specific interactions have been definitively identified in clinical practice. More commonly, clinicians need to be aware of potential functional interactions, additive effects with other substances that cause sedation or dizziness, such as benzodiazepines, opioids, certain antipsychotics like quetiapine, and of course alcohol. Caution is needed when co-prescribing. Nabilone is not approved for pediatric use, and again, while there's been interest in its use for agitation and dementia, it's not formally approved for this.
How is it typically dosed?
In clinical use for CINV, nabilone is usually started at a dose of 1 or 2 milligrams, given 1 to 3 hours before chemotherapy administration. Sometimes an initial dose is given the night before chemo to help prevent anticipatory nausea. Dosing can be continued every 8 to 12 hours during and after chemotherapy, typically for 24 to 48 hours, but the total daily dose generally shouldn't exceed 6 milligrams. Since side effects are dose-dependent, starting with 1 milligram is often recommended, increasing to 2 milligrams only if needed. For off-label uses, like controlling PTSD-related nightmares, nabilone is typically taken about 1 hour before bedtime, often starting at a low dose like 0.5 milligrams and gradually titrating upwards, perhaps to a maximum of 4 milligrams per night, depending on individual response and tolerability. Similar dose ranges might be explored for chronic pain or agitation, always starting low and going slow.
Okay, let's shift our focus now towards the future of cannabinoid drug development. And we have to start with a really fascinating and somewhat cautionary tale that profoundly shaped the entire field. This feels like a classic, here's where it gets really interesting, moment in the history of drug development. A therapy that showed immense promise but ultimately hit a major wall due to unforeseen side effects. It perfectly illustrates just how complex it is to target a system as widespread and fundamental as the ECS. We're talking about the effort to develop peripherally restricted CB1 antagonists.
The story truly is a pivotal chapter, a crucial lesson learned for anyone involved in pharmacology or drug development in this space. The motivation behind developing peripherally restricted CB1 antagonists was initially very strong and logical. There was compelling preclinical data suggesting that blocking CB1 receptors only in the periphery and tissues outside the brain and spinal cord could offer significant metabolic benefits.
Like what kind of benefit?
Things like weight loss, improved insulin sensitivity, better glucose control, addressing key aspects of metabolic syndrome and type 2 diabetes. The hope was to achieve these benefits without the psychiatric side effects like anxiety and depression that had been observed with earlier, non-selective CB1 blockers that did enter the brain. The underlying assumption at the time was that those negative mood effects were solely due to the drug acting centrally in the brain. So the clear development goal became create CB1 antagonists or inverse agonists that don't cross the blood-brain barrier, keep them out of the brain. And this brings us directly to the story of rimonabant, which is detailed in box 26-1 of our source material and serves as a critical case study. Rimonabant was identified by Sanofi Aventis. It's technically an inverse agonist at CB1 receptors, meaning it doesn't just block activation, but actively reduces the receptor's baseline activity. The rationale was that by blocking CB1 receptors, particularly those involved in appetite regulation, it could decrease the consumption of highly palatable, rewarding foods and serve as a novel treatment for obesity.
Did it work in clinical trials?
Yes, in clinical trials, rimonabant consistently produced modest but statistically significant weight loss across diverse groups of obese individuals. But what was particularly intriguing, and a key finding, was that it also improved various metabolic parameters, things like blood glucose levels, insulin sensitivity, lipid profiles, often to a greater extent than could be predicted simply from the amount of weight loss achieved.
So it wasn't just about eating less.
Exactly. Further investigations suggested that the weight loss induced by rimonabant wasn't primarily due to a sustained decrease in food intake. That effect seemed to be somewhat transient, wearing off after the initial period of treatment. Instead, a significant part of the sustained weight loss was attributed to an increase in energy expenditure. This was thought to result from rimonabant directly antagonizing CB1 receptors located on peripheral tissues like adipocytes, fat cells, hepatocytes, liver cells, and skeletal muscle cells, influencing their metabolic activity.
So it seemed very promising initially. What went wrong?
Based on these positive weight loss and metabolic data, rimonabant was approved for use as an obesity treatment in the European Union. But and this is the crucial turning point, soon after it hit the market, serious concerns began to emerge from post-marketing surveillance and ongoing clinical studies. These concerns centered on a significantly higher risk of psychiatric adverse effects, specifically anxiety, depression, and tragically, an increased risk of suicidal ideation and behavior in patients taking rimonabant compared to placebo.
That's incredibly serious.
Absolutely. Because of these severe psychiatric safety concerns, rimonabant ultimately failed to gain SEA approval in the United States, and shortly after its European launch, it was withdrawn from the market there as well. The rimonabant story had a profound and lasting impact on cannabinoid drug development. It dramatically highlighted the potential dangers of globally blocking CB1 receptors, even if the primary target was peripheral metabolism. It strongly shifted the research focus towards developing truly peripherally restricted compounds with meticulous testing to ensure they don't cross the blood-brain barrier, or exploring alternative strategies altogether. It taught the field a hard lesson about the intricate links between peripheral and central ECS function and the critical role of CB1 in regulating mood and emotional well-being, underscoring the challenge of disentangling its metabolic roles from its central ones.
So, if directly blocking the CB1 receptor globally proved too risky due to those central side effects, what other strategies are researchers pursuing now? It seems like targeting the enzymes that break down our natural endocannabinoids, FAAH and MEGL, might be a more subtle and potentially safer approach. The idea is to boost our own internal signals rather than hitting the receptor directly with an external drug, right?
That's exactly right. This strategy represents a major and arguably more nuanced approach currently being explored in cannabinoid drug development. Instead of using exogenous agonists or antagonists that directly interact with the cannabinoid receptors, the goal here is to selectively inhibit the catabolic enzymes responsible for degrading the endogenous cannabinoids AEA and 2-AG.
Why is that potentially better?
The thinking is that by gently inhibiting the breakdown of AEA or 2-AG, you can naturally enhance their levels and duration of action, specifically where and when they're being produced physiologically in response to demand. It's about potentiating the body's own signaling, allowing the endogenous system to operate more effectively, potentially offering therapeutic benefits with fewer off-target effects or the risks associated with global receptor blockade or activation. So specifically, FAAH inhibition aims to potentiate AEA signaling, and MEGL inhibition aims to potentiate 2-AG signaling.
How are these approaches progressing in clinical trials? Let's start with MEGL inhibitors.
MEGL inhibitors have entered clinical testing, pursued by several pharmaceutical companies, although progress has faced some hurdles. For instance, an early compound developed by Pfizer did not advance past phase I trials, reportedly due in part to challenges with achieving adequate brain penetrance. Another MEGL inhibitor, initially developed by Abide Therapeutics and later acquired by Lundbeck, known as LUGI-AG06466 or ABX1431, unfortunately proved ineffective in a clinical trial for Tourette syndrome.
So setbacks there. Is there still promise for MEGL inhibitors?
There is still significant future promise, particularly given MEGL's unique role in neuroinflammation. Remember how MEGL activity, especially in astrocytes, controls the liberation of arachidonic acid from that large 2-AG pool, which then fuels the production of inflammatory prostaglandins via COX2. This specific link makes MEGL inhibition an attractive strategy for conditions where neuroinflammation is a key driver. Consequently, ongoing clinical studies with MEGL inhibitors are increasingly focusing on neurodegenerative diseases, such as Alzheimer's disease or perhaps Parkinson's disease, where modulating central inflammatory processes could be beneficial. The ability to both boost neuroprotective 2-AG signaling and reduce the production of inflammatory molecules from its breakdown makes MEGL a compelling target in that context.
What about FAH inhibitors? Have they seen more testing?
Yes. FAAH inhibitors have generally undergone more extensive testing in clinical trials compared to MEGL inhibitors, and they've showed some intriguing, though sometimes inconsistent, results. The first FAAH inhibitor to enter broad clinical development was PF04457845, developed by Pfizer. This compound is a covalent, irreversible inhibitor, meaning it permanently binds to and inactivates the FAAH enzyme. Studies showed it could produce remarkable up to 10-fold elevations in circulating levels of AEA and related fatty acid mides, sustaining these elevated levels for a full 24 hours. Importantly, initial safety studies indicated good CNS penetration, effective saturation of FAH enzyme activity in the brain, and crucially, no major serious adverse side effects were detected.
Sounds promising. Did it work for its initial target?
Well, its first large-scale clinical trial was for osteoarthritic pain, and surprisingly, it failed to show efficacy compared to placebo. This was a significant setback and led to some rethinking about the primary role of AEA in modulating chronic pain of that type. However, the story for PF04457845 didn't end there. In 2019, a phase TIA trial demonstrated both safety and efficacy in treating symptoms of cannabis withdrawal and dependence in men. Patients receiving the FAAH inhibitor showed notable improvements in mood disturbances, anxiety, and sleep changes commonly associated with cannabis withdrawal. This positive result has led to a larger multi-site trial which is currently ongoing, suggesting real potential for FAAH inhibition in addiction medicine.
Any other positive signals?
Yes. Further bolstering its potential in stress and anxiety, an experimental trial in healthy humans published in 2020 showed that PF04447845 significantly reduced effective autonomic and physiological responses to laboratory-induced stress. It also improved fear extinction learning, a process critical for overcoming anxiety disorders. Building directly on these findings, studies are now actively underway examining the efficacy of FAAH inhibition, specifically in patients with post-traumatic stress disorder, PTSD. Other FAAH inhibitors have also been tested by different companies, adding more pieces to the puzzle. For example, J&J 421-65279, a reversible FAAH inhibitor developed by Janssen, was found to be ineffective in a trial for anxious depression. However, interestingly, the same compound did show some evidence of benefit in patients specifically with social anxiety disorder, hinting again at a potential role in certain anxiety subtypes. Another FAH inhibitor, SSR411298, was tested for geriatric depression, but unfortunately showed no significant benefit.
So what's the overall outlook for these enzyme inhibitors? Where might they fit in therapeutically?
The overall clinical utility of these endocannabinoid modulating drugs, both MGL and FAAH inhibitors, is still being determined. It's clearly not a simple magic bullet approach. However, specific areas of promise are emerging. MGL inhibitors continue to hold potential primarily for neuroinflammatory and neurodegenerative diseases, leveraging that unique link between 2-AG metabolism and brain inflammation. FAH inhibitors, based on the current evidence, seem most promising for treating stress-related psychiatric disorders, such as PTSD and potentially social anxiety disorder, as well as potentially aiding in the management of cannabis use disorder. This really raises a crucial question for the future of pharmacology in this field. Will subtly modulating our own internal cannabinoid levels by targeting these enzymes prove to be the key to unlocking new, effective treatments, offering a perhaps more physiological and balanced approach? Or will the inherent complexity and widespread nature of the ECS continue to present formidable challenges, requiring even more precise targeting and context-specific interventions to achieve therapeutic success without unintended consequences? It's a fascinating area to watch. #outro.
Wow, what an absolutely incredible deep dive that was. We have really unpacked so much today, from the elegant, intricate, on-demand signaling of our body's own messengers, AEA and 2-AG, right through to the profound and often complex ways that compounds like THC, CBD, and even those dangerous synthetic cannabinoids interact with this fundamental system. We've seen how it touches almost everything.
It really is astonishingly widespread. If we connect this all back to the bigger picture, truly mastering this information means appreciating the profound interconnectedness of our body systems. You, our listener, now have a foundational understanding that goes far beyond the usual surface-level discussions. It positions you to approach conversations about cannabis, about pain management strategies, mental health treatments, metabolic disorders, and future drug development with a genuinely informed perspective, thinking about the mechanisms like an elite professional in the field.
Absolutely. So what does all this mean for you? Maybe take a moment to consider how this incredibly intricate system, so deeply woven into the fabric of our biology, might continue to surprise us as research inevitably unfolds. What other hidden connections, what other subtle regulatory roles might the endocannabinoid system be quietly orchestrating within us, just waiting for the next generation of scientists to discover?
Indeed. The story of the endocannabinoid system is clearly far from over. Its complexity and ubiquity encourage scientists and the informed public alike to constantly question our assumptions, to remain curious, and to explore the full spectrum of possibilities held within our own physiology. It's a powerful reminder of just how much more there is to learn about the remarkable biological systems that make us who we are.